Oui, il est fou, mais en fait, ça n'a rien. On va pas se demander on va pas demander
Je pense que
voilà. Ça, c'est l'avantage d'avoir une bonne formation. Alors que il y en a un, on tombe sur du coup, ça va bien. Trop de travail. Alors, on va y arriver. Oh, yes.
I have another class at 5:45, 3:45 p.m. at university facility. So I really have to leave at 10 minutes, but no more. Okay, so I will begin by check who is present. So Emanuela. Okay. We need to do this. Oui. Samuela, passé. Elles sont ben non, on ne les a pas marquées. Ok. Ah, mais elles sont là. I see you. Donc, Isabella, partage. Ah oui. Il est venu aussi dans le. Ok, Janine, Marianne, ok, Alain, yes, et Luna, elle a fait ça à 14h. Ouais, je sais bien. Et Maïa, oui, ok. Alors, Sophie, et Hélène, ok. Mana, elle arrive aussi. Okay. Antoine, Mania. So, Mana is in Toulouse pour l'instant. Okay. Elle part vendredi. Ah, but quiet. Et Yamina. Okay. Antoine. Émilie, pardon. Raphaël. Oui. Rania. Alexa. Léo. Léona. Anaïs. Serena. Oui. Coraline. Oui. Léa. Oui. Guillaume. Oui. Jade. Oui. Chloé. Oui. Donc, Lola, c'est bon. Lisa. Oui. Maëlina. Oui. Et Elisa. Oui. Et ensuite, donc, Coralie. Vincent, oui. Benjamin. Solène. Oui. Déborah. Oui. Léa. Oui, je l'ai vu aussi. Lucas. Aussi, je ne me rappelle plus les prénoms. Mélanie. Morgane. Oui. Oui. Fatima. Chloé. Jules. Oui. Iman. Oui. Pauline. Guillaume. Oui. Chloé. Colia. Oui. T'as pas de jalousie avec ton voisin. Et Nina. Oui. Um, Today we'll speak about 3D models and their application in preclinical evaluation, mostly in cancerology. So first, we will start with the two presentations of the articles. And then I will give some compliments to the papers to highlight some specific aspects associated with 3D models and their use. After the presentation of articles, please ask questions. I'm sure you will have many questions. Not everything will be fully clear. So take part of the discussion. This is really for you. So use it. Okay. In Zoom, is it okay for the sound? Yes. No.
Yes, it is.
Okay, great. Thank you. So who wants to start? We start with the endometriose, perhaps. Is it okay for you?
Après, vous avez dit que vous avez dit la Ah, moi aussi, je vais me demander pour la manipulation. C'est ça,
So we would like to share with you an article published in 2019 in the scientific journal Nature Cell Biology and entitled Patient Derived Organoids from Endometrial Disease, Clinical Heterogeneity and are Able to Drug Screening. So first of all, an introduction about the endometrium, which is the mucous membrane that lines the inside of the uterus. It's made up of several layers of tissue rich in blood vessels and glands. And its main role is to create a favorable environment for the implantation of an embryo during the fertilization. During the menstrual cycle, the endometrium undergoes changes in response to hormones, in particular estrogen and progesterone. So the endometrium is essential for fertility, and abnormalities can lead to disorders such as endometriosis and endometrial hyperplasia. So endometriosis is when an endometrial cell develops outside the uterus, for example, on ovaries or on fallopian tubes. It affects 10% of women on a childbearing age. And the endometrial hyperplasia is characterized by an excessive thickening of the endometrium, which is often caused by a prolonged estrogen stimulation. And this situation can lead to a potential endometrial cancer if left untreated. So the endometrial cancer is the most common gynecological cancer in the middle-class countries, notably for women, post-menopausal women. And there are two types of EC. The type 1, which is estrogen dependent, it affects women between 50 and 60, and it represents 80% of cases. But it's associated with a favorable prognosis, and the overall survival at 5 years is near 85%. And then there is a type 2 EC, which is estrogen independent for women over 65 years old, and it represents only 20% of cases, but associated with a poor prognosis. And finally, the overall survival at 5 years is between 25 and 60%, and it's associated with the high grades and high risk of metastasis. So there are main types of treatments. For example, the surgery, and the treatment depends on the stage and the type of cancer. The surgery, which consists of an hysterectomy, which is the removal of the uterus. There is also the bilateral salpingo oophorectomy, which is the removal of ovaries and fallopian tubes. It can be accompanied with radiotherapy, and that's reduce the risk of relapse. There is also chemotherapy for advanced stage and the type 2 cancer, which are estrogen independent, but also in the event of relapse. And finally, we have the hormonal therapy, mainly for type 1 cancer, which is estrogen independent. And it acts by blocking the action of estrogen. And finally, the immunotherapy, which are new approaches, notably for tumor with the microsatellite instability. But despite all of these types of treatments, the efficacy of therapy and overall survival rates are not substantially improved. In addition, we don't really know the pathobiology of this disease.
So to improve our understanding of endometrial cancer, 3D models can be developed in order to reproduce the complexity of the endometrial structures. For example, by mimicking the interactions between cancer cells and their microenvironment. So unlike conventional 2D cell culture, where cells are grown in monolayers, 3D models actually allow the cells to develop in a three-dimensional environment, which is closer to what we can find in vivo, for example, in the uterus. So those 3D models include spherical spheroids. Spheroids are a cell that aggregates as a spherical structure and that can autonomously organize themselves. They are mainly used to study cell growth, signaling, response to treatments, but also oxygen and nutrients gradient that we can find in vivo. We also have organoids, which are 3D models derived from embryonic or adult stem cells that are able to self-organize in order to mimic mini organ. So they can be used to model specific organs like brain, lung, and also the liver, for example. But they're also used to study genetic disease as well as drug response. And they can also be derived from cancer cells. So in order to test specific treatment. There is also organ on chip. So organ on chips are miniaturized microfluidic devices that contains living cells and which will replicate the function of an organ once again. So they can be done with microfluidic channel, which can simulate the flow of body fluids, like the blood, for example. They are mainly used to study pharmacokinetics as well as response to toxins and the progression of disease because of their similarity with the dynamic of the organ. However, current 3D models actually fail to reproduce the nature and the heterogeneity that we can find in endometrial disease. So this limits the clinical but also scientific progresses that can be made. So despite the advances, creating a 3D model that can replicate all of this is still a complicated task. Indeed, it is known that the few cell lines that are derived from carcinoma are not able to display all of these heterogeneity that we can find in endometrial cancer. Furthermore, the grade type 1 tumors are not able to grow correctly in vivo nor in vitro nor in vivo as patient-derived stem grafts. And finally, there are no study models for precancerous endometrial hyperplasia, and the animal models are not able to display the typical human disease. So the challenges here are to develop, to reproduce the hormonal environment of the menstrual cycle, but it is also to develop a model that can reproduce the heterogeneity that we can find in endometrial tumors, as well as the interactions with other cells in the body. So these are the challenges that the authors wanted to address in this article that we are going to present.
So the first aim of the authors was to create a long-term expandable organoids from endometriotic lesions. So to remind you, endometriosis is when the endometrium-like develops in ectopic sites, such as the peritoneum and the uterus. And in order to make this organoid, they used endometriotic samples with the optimized culture medium, and they amplify them in order to analyze them and make the biobanking. So first, they observed that ectopic organoids named ECTO develop between 7 and 40 days, but in smaller numbers than organoids from healthy endometrium named EMO. And in addition, they also observed a lower efficiency of ectopic organoids, which can be due to several dissociation needed for ectopic endometriotic tissue. After that, they also made a quantification of the fluorescence emitted by the transcription factor Ki67, which is a marker of proliferation. And they observed the significant proliferation of ECTO, which were able to be extended over the long term in the same way as EMO. As we see, there are also ECTO even with 18 passage, which represents 8 months, and even with a number of passages. And finally, they also make an analysis of the gene transcription of gene expression of endometrial markers, and they show that ECTO showed transcriptomic stability during long-term expansion, even with the high passage. And they conclude that long-term expressible organoids can be established from endometriosis.
So after that, the authors wanted to know whether ECTO could recapitulate the phenotype of endometriosis, which include, for example, endometrial lesions. So just as a reminder, in endometriosis, there is endometrial cells that can migrate out of the uterus to deposit in other organs in the abdomen, which can trigger inflammation, but also lead to scars and lesions. So here, by histological analysis, the authors studied the structure of the ECTOs and compared it with the reference, which is endometriotic lesion here. And what they could observe is that the ECTO showed a luminal invasion, which was also consistent with the expression of multiple mechanistic luminance. So we can see on the right. Then they did an immunohistochemical analysis in order to check the presence of endometrial markers, such as the presence of estrogen receptors, but also progesterone receptors. And indeed, we can see that both receptors are conserved in the organoids. They also realized periodic acid shift staining, which is PAS, to detect the presence of mucin. And we can see that both the primary endometriotic lesion as well as the organoids secrete the mucus into the lumen. So this last result actually suggests the presence of secretory cells, which was confirmed by the presence of microvilli and cilia that are directed towards the lumen in the organoids. So this was for the in vitro part. Now the authors wanted to know if this was also the case in vivo. So to do this, they realized an ETTO graft by subrenal transplantation in ovariectomized non-skin mouse, and they observed the endometrial markers such as the receptors once again, and they saw that indeed the implants conserve the expression of hormone receptors. However, we know that endometriotic lesions are usually located in a more peritoneal area. So they did a second experiment that is illustrated on the left, where they grafted ECTOs in the peritoneal cavity of ovariectomized non-skin mouse. And they also did a control with a sham non-skin mouse. So they let the implant for 2 months, and they observed that indeed the implant was expressing the endometric markers like the receptors or even the presence of mucin. So thanks to all of these results, the authors were able to conclude that ECTOs did indeed summarize the phenotype of endometriosis, not only in vitro, but also in vivo.
So then the authors wondered if ECTO has genetic and transcriptomic characteristics associated with the endometriosis. And to answer that, they performed the RNA-seq and the heat map on ECTO, UTO, or for atopic endometriosis, and EMO, and also performed the diagram, which showed that there is a presence of differentially expressed gene specific to the type, and within the same organoids themselves. So after this, they also performed the gene ontology analysis, which compared ECTO versus EMO in order to find the pathway altered, and they find that interaction between the extracellular matrix and receptors, but also some signaling pathway like the Wnt pathway and the PE3 AKT pathway, but also the adhesion mechanism and invasion were altered. And this last point, the adhesion mechanism and invasion, were important for the tissue anchorage and also the growth in the ectopite sites. So this one is associated with endometriosis. So the comparison between ECTO and EMO allowed the authors to know what pathway are affected in the endometrium of patients with the endometriosis. So they performed the RT-qPCR that quantified the expression of genes coding receptor of the Wnt pathway. And to remind you, Wnt beta-catenin pathway is involved in the cell proliferation, and it's often overactivated in cancers. They made this experiment on different stages of ECTO. They compared with the EMO and then find the upregulation of LEF1 and LGR6, so an upregulation of the Wnt pathway in the ECTO. And they also make an RT-qPCR in order to quantify the gene involved in inflammation and inflammatory. And endometriosis is associated with inflammation, so they wanted to confirm that on the different ECTO. And they find that metalloprotease-2 and inflammatory cytokines were upregulated in the different stages of ECTO. And they finally conclude for this figure that transcriptomic and genetic analysis of ECTO revealed disease-associated characteristics.
So after that, the authors wanted to develop organoids from precancerous endometrial tissues. So in order to do that, they used hyperplastic endometrial tissue, as hyperplasia can lead to the development of cancer. So they did those organoids that are called EPO, and it resulted in an efficiency of 70%. So for those organoids, actually 3 subtypes of tissues were used: simple benign, complex atypical, and polyp. So the simple hyperplasia is actually characterized by crowded glands as well as irregular shape and size, but the cells do not appear atypical. Whereas in the complex hyperplasia, we have the same characteristics, so the cells do appear atypical. So the organoids were done with these three different types of tissue, and the authors were able to conserve them in the long term for more than 6 months, despite the high passages you can see here. So the first analysis that was done on these organoids was the quantification of fluorescence from Ki67 positive cells, which showed a very high proliferative activity in each of the three type of organoids. The authors then looked at the molecular and subcellular characteristics of the tissues, once again, like the receptors, the presence of mucin, and so on. And they realized that it was all equally reproduced in the hippo models. Finally, they performed targeted sequencing analysis as well as the genomic analysis on primary biopsies of Lynch syndrome patients as well as their corresponding organoids. So just to introduce Lynch syndrome, it's an hereditary predisposition to cancer, which primarily develops in the colon and in the endometrium. So usually people who have this syndrome have mutation in and so that's what we're looking at here. Up we have the primary tissues, and under we have the corresponding organoids. And the authors noticed that specific mutations that were affecting the mismatch repair genes, MSH2 and MSH6, were conserved in the organoids. So thanks to all of these results, the authors concluded that hippo, which are organoids developed from precancerous endometrial pathologies, did capture the phenotype as well as the genetic mutation of the disease.
So the finally aim of the authors was to create organoids derived from endometrial cancer, and they wanted to know if these organoids represent the phenotype of the disease. So they use different samples from tumor, EC tumors, and from different grades. They first observed that the ECO developed in 7 to 20 days, but the efficiency was way lower, only 20% than the other endometrial conditions. And also organoids showed a limited expansion potential. So by finding the important elements for the development of the organoids in the medium culture, they improved the ECO efficiency after optimization, and they also improved the development for more than 8 months. So in other words, the organoids lasted over the time. After that, they also make a quantification of the proliferation and the number of, they found the number of organoids per well, and they saw a strong proliferative activity and phenogenic capacity of ECO, which is also a characteristic of the endometrial cancer. They also observed a morphological heterogeneity between the different kinds of organoids, and indeed they are from different types of patients, and also you have a heterogeneity between them. So that can be explained by the morphological heterogeneity, which is also a characteristic of endometrial cancer. And finally, they compared samples of endometrial cancer and organoids by staining and immunohistochemistry. And they saw that ECO replicated the histology of the primary tumor with the expression of different markers associated with endometrial cancer. And in addition, we also observed an expression of estrogen and progesterone receptors, which are maintained in the organoids. And finally, it is known that the PE3-AKT pathway is frequently hyperactive in endometrial cancer. So the authors wanted to confirm that in the different ECO organoids, primary, late, and early, by using a mutation matrix, which refers to different mutations that affect the endometrial cancer. And they find notably genetic alterations in the pathway signaling mediators, notably PTEN, multiple line of ECO. And to remind you, PTEN is a negative regulator of the PE3-AKT pathway. And finally, this conclude with the ECO capture the mutational and structural landscape of the tumor and display a gene expression that is specific to the disease.
So after that, the authors wanted to know if ECOs could reproduce the phenotype of the disease by the time in vivo. So to do that, they performed a subcutaneous injection of ECOs in not-skilled mice. And what they could see is that ECO was able to generate a cell mass that reproduced not only the histological, but also the molecular characteristics of the primary tumor. So After that, they also performed orthotopic grafting of material suspended ECO in uterine horn, in the uterine horn of non-skid mice. So what they observed here is that, so there was two results, one for high grade and one for low grade ECOs. For the high grade ECO, there was a highly proliferative and invasive mass, with also the presence of metastasis in the peritoneal area. And in contrast, for low-grade ECO, the lesions really remained located in one point, and there was less proliferation. Also, it's important to notice that the orthotopic grafts were also able to reproduce the hormonal phenotype with the presence of estrogen and progesterone receptors, just like in the original cancer tissue. So here, the authors were able to conclude that the ECOs that are developed from endometrial cancers were able to reproduce the disease, the phenotype of the disease in vivo. Finally, the authors wanted to know if those organoids were suitable for drug screening. So in order to do that, they tested 4 different chemotherapeutic compounds that are listed here on the right, as well as an mTOR inhibitor, which is everolimus. So they did all of the testing, and they observed that the organoids were indeed suitable for drug screening, as they present patient-specific drug response. It is also important to highlight the fact that one of the ECO2, was actually the most sensitive to everolimus, which can suggest a high dependence on the PI3K-AKT pathway, which is also in accordance with the previous results. Like we saw, there is mutation in the mediators of this pathway, like for example, PTEN. So in conclusion here, the ECOs have patient-specific drug responses and also represent the heterogeneity that we can find in endometrial tumors.
So finally, the endometrial disorders are a major gynecological problem and a cause of infertility. So the authors wanted to develop the organoids that cover a broad range of endometrial diseases, such as endometrial cancer, endometrial hypoplasia, and also endometriosis. So these organoids capture the heterogeneity of the disease, maintain the characteristics of the primary tissue, and also have a strong expansional ability, so long-term use, that can decrease the problem of limited primary biopsy and decrease the animal experimentation. And these organoids are a beginning of a biobank, which cover healthy and pathological endometrium, which can be a tool in order to search new mechanisms and drug targets in the field. So about the limits, we can say that the organoids can be an incomplete tissue complexity because they are mainly composed of epithelial cells. So they don't really reproduce the whole cellular diversity, notably the smooth muscular cell, the stromal cell, and also the immune cell and the vascular cells. So we don't really reproduce the cellular interaction and the immune response, which is important for the regulation of the endometrial cancer.
So there are also difficulties to reproduce the menstrual cycle. As we know it, the endometrium is quite unique tissue in the way that it is subject to cyclic variations due to the influence of hormones like estrogen and progesterone. So reproducing this complex environment and its effect on organoids is still quite complicated. So to this day, the organoids don't fully capture the dynamic of the menstrual cycle. There's also a lack of blood vessels as well as nerves, which are essential to study hormonal regulation, but also inflammatory response. So these, and notably the blood vessels are actually key to understand the processes involved in tissue growth as well as the development of metastasis in endometrial cancer. So the absence of all these elements can actually limit the accuracy of the representation, whether we are studying a physiological process or a pathological process. And finally, there are definitely a lack of microenvironments. As we know it, organoids are developed in very stable and controlled environments, which do not necessarily reflect the complexity that we have in vivo with the endometrium, since we have mechanical factors, but also biochemical factors and interactions with the surrounding tissues and the cells, like the immune cells that we cannot find in immunodeficient mice. So all of these elements limit the study of interactions with the microenvironments in the study of things of mechanisms by invasion, immigration.
Other question? We first listen to you.
The optimization way to 20 to 40% in you. We the authors by different type of cultural medium. They find important factors such as IGF-1, the insulin growth factor, and they also use an inhibitor of P38, and they saw that the efficiency was greater than before. So they use a new type of medium in order to have the organoids.
And we talk about sound and there is a graph with sound.
I understand one of the first experiments when they took ACT organoids, they implanted in ovarian-optimized mice. And the extension was the same with the control mice.
So yes, they implanted this one.
No, before with the ovarian-optimized mice. And it's
We're talking about the same segments. Because what they did is that they implanted ECTOs, so in the peritoneal cavity of mice. They did it for 2 months, and then they observed the expression of the markers that we are supposed to have in vivo. And for the control mice, what they did is it's a sham not skid mouse, so they performed the surgery, but without actually implanting the ECO. So it's just a control to check if the surgery is not actually affecting the results.
my question is why they use an ovarian-optimized mouse.
So I think it's mainly to have like standardized mice, because what they did actually after writing the CTO is that they also used estradiol pellet. It's like a little stock of estradiol that will release continuously the hormones. So we have a more stable hormonal concentration instead of having variations. And that's why they like to make
Exactly my question.
You talked about the microenvironments, but do you know, I don't know, in article, if the author are thinking about that, to put microenvironments in this experiment or not?
No, they don't think about add microenvironment. They also make the organoids with only epithelial cells. So they want to have different type of cells which are involved in the microenvironment.
Other questions? About microenvironments, there's at least some aspects that are included in the culture of organoids. What did they use from the culture? Medium, of course, but there is also another.
They use a matrigel with growth factors.
Yes, and you all know what is matrigel, I guess. Yes. Yes, explain to me. What's matrigel? There, okay. And then.
Extracellular proteins.
Extracellular proteins, but of what? From the animal? No,
They need the matrix.
yes, it included protein of the extracellular matrix. And matrigel is not a synthetic matrix. It's from animals, but not from mice. Okay. And one problem with the matrigel is that there is a problem of stability and reproducibility between different
Different conditions.
not different conditions, but when you buy a material, you ask for a loan, and then when you buy another, it can be different, and you can have some consequences on the reproducibility of the culture. But no, the point is the extracellular microenvironment for epithelial cells, and in material it is also enriched in laminin. Laminin is part of the basal lamina. Thank you. La basal, the basal lamina. Okay. That is with which integrins of the epithelial cells interact. Okay. So this is really important in the production of organoids or other kind of 3D models. And the first works on 3D models were performed by Nina Bissell quite long ago now. But the experiments is based on the use of mammary epithelial cells. Some are normal cells and some are cancer cells in the experiment. And when they culture cells in 2D, they do not see any difference between the normal and the cancer cells. And when they put these cells in this specific matrix, no, so enriched in laminin, this is really important. They do see a difference between a normal cell that reproduces acinus with production of proteins that are secreted, and cancerous cells form the irregular aspects with epithelial cells lose the epithelial cell polarity and so on. And it is only in 3D culture that they can see the difference between normal and cancer cells. Okay, and that's the starting point of the use of the material in many 3D minerals culture. And in organoids, it's really important. So the material, and you also speak about the growth factors in the medium, and in the paper, they show that first figure, I will remember, they test the composition of the growth medium. So it is true at the end to optimize the medium of the cancer models, but it is also true at the beginning for the non-cancerous models, where they tested different growth factors in the medium. And this is also really specific for the organoid culture to really optimize the culture medium with specific growth factors. And for each kind of organoid, there is a specific medium. So the consequence of this is that the culture medium is quite expensive in a way, because we have to complement with different growth factors, and these growth factors are quite expensive, as well as nutrition. So it's not so easy to obtain organoids in culture. So for normal tissue, but also for cancer tissues. This is one aspect. About the complexity of the model and the idea to be able to increase the proximity of the model with the reality in vivo. What's the purpose? For the 3D models, the objective is to not to work on 2D cells that are plated on a plastic dish that is really hard for the cells without any cell-cell interactions. Okay. And in this, it is not yet the case, but there are many work on organoids model or on spheroids model, as you presented in the introduction, where they tried to add in the environment immunologic cells, fibroblasts, tumor-associated fibroblasts, and they are like this mixed model that are more and more used and published with these 3D models. As well for the presence of the vascularization. This is also a problem, you're right. And there is here also different works, mainly in collaboration with physicists, to be able to have some structures resembling vessels inside the 3D models. So in some models, not really in organoids, it is more models that are closer to spheroids. They can drive UV cells to form structure that looks like vessels in 3D. There are some work on this, but it is not yet totally general, not at all. Organoids today, there are organoids for everything. Organoids, the first paper on organoids, it was in 2007. The first model is on colic epithelium, sorry, intestinal epithelium. It was associated with the discovery of stem cells in the crypts of the intestinal epithelium. And since then, so in 15 years, something like this, now there are organoid models for quite every organs in physiological and pathophysiological situations. So, and we found organoids in every lab. I think there is no lab without organoids today. But depending on the research activity, there are models that are more or less represented, but they exist everywhere. And so in in the context of cancer, but also in developmental biology, also in regeneration, and every biological question. But it is not easy to obtain organoids, and we will talk about it later on. But it is difficult. It is not discussed. I don't think we don't. But I think it's not discussed in the paper to have really reproducibility in the size and the shape, in the percentage of the heterogeneity in cells. Because the advantage here is that these models reproduce the cell heterogeneity found in tissues or in a pathological situation. So for tumors, for example, heterogeneity of cells in tumors is maintained in organoids with the diversity of genetic alterations found in patients. So this is really important and really interesting, but to have it to be in conditions that are that can be applied for pharmacological evaluation, for example, it is not yet the case today. There are many research papers. There are development of organoids in the industry, but it is not yet totally used for evaluation of new molecules for high-throughput screening, for example. Okay, but we will discuss this in more detail just after. Other questions, remarks, comments? No? We will listen to the next presentation. It will be okay, don't worry. We have this in two parts. It will be on glioblastoma. Generally, in glioblastoma. So the first part will not be very close to 3D model, but we'll have a complementary action. As you prepare to do this, because in the presentation of the I guess an introduction small detail, yeah, and then move in order you have to use the because if not it's not last question is moving topic. Can you the chat? No, no question. Okay. Okay, we listen to you.
We will present to you the review which entitled circular arena novel types of biomarker for bioma. First of all, we talk about cells, bioma cells. The main role of the cells is to support and protect neurons. Regarding malignant tumors, these are primary tumors of the central nervous system, accounting for more 75% of malignant tumor in the brain. Bioma tumor can be classified based on the different type of cellular we will see this week. There are many treatments with radiotherapy, chemotherapy, and surgery, but the disease often remains persistent or recurs. Early detection of biomas can be difficult due to the deep location and the lack of symptoms. And by the time symptoms appear, the cancer is already in the advanced stage. The future is therefore can circular RNAs provide a more effective and stable alternative to current biomarkers for the diagnosis, prognosis, and treatment of biomas, and what challenges remain for the clinical application. Circular RNAs are non-coding RNAs, meaning they do not code for proteins. There are three types of RNAs, like microRNA, long non-coding RNA, and circular RNA. And the circular RNA have many roles, like regulatory functions. The conformation provides protection against RNA, and they have various combinations of sequences and longs.
For better understanding, we try to present you an example from this review that shows us when there is a dysregulation of the circular RNA's quantity. Here, lack of circular RNA. There are impacts in the progress and in the proliferation of the glioma. And for example, for this example, they took all these circular RNA, which are tumor suppressors. So you can understand that if there is less tumor suppressors, then physiologically there is impact on the proliferation. So more proliferation, invasion, metastasis, angiogenesis, and less apoptosis. So we can say that circular RNAs are key modulators of tumor formation. As I said, abnormal circular expression, it can be an augmentation or a decrease of this quantity, induces modulation of glioma migration, apoptosis, proliferation, invasion, and angiogenesis, mainly by acting as mRNA targeting sponge. But what is that? In fact, the circular RNA do not have a direct impact on the expression of the gene, whereas mRNAs do. So when the circular RNA targets the mRNA, it can't play their role on the expression of the gene. So for another example, you have an mRNA here that have an inhibition of the traduction on the proliferation and avoids the expression of the potential genes. And if there is a circular RNA that captures this mRNA, you will have a lot of traduction, proliferation that will impact in tumorigenicity in glioma cells, and the same for the potential genes. That's why research on circular RNA in gliomas is paving the way for new therapeutic approaches based on the modulation of circular RNA expression.
We will talk now about circular RNAs as diagnostic and prognostic biomarkers in glioma. Early detection of gliomas can be difficult due to the location and the lack of symptoms, and this is why circular RNA can be used as biomarker, as prognosis, for glioma is poor compared to the other types of cancer. Circular RNAs have a close ring structure, which stabilizes circular RNAs in plasma and tissue and protects them from the enzymatic activity of exonucleases. Oops. And all of this allow potential advantage for their use as biomarkers for the diagnosis and prognosis of glioma. We will talk now about therapeutic potential of circular RNA in glioma. There is chemotherapeutic treatment and surgical injection. But they are often followed by cancer reliefs. There are an example in the review. By the activation of circular RNA, which entitled NFI8, we can achieve the inhibition of the invasion inside apoptosis in glioma cells, the inhibition of the cell migration and cell cycle arrest, and all of this allow to construct tumor.
So I took this article from this review and I choose some results to show you. So the scientists works on 2 different cell lines in glioma, so 2 different people, and they in-silence the NFX, circular RNA, and here we are looking at the relative glucose consumption, and we can see that the NFX silence significantly inhibited glycolysis in both their line, revealed by a reduction in glucose consumption. This is directly linked with the activity of the cells. So we can see here with the in-silence, there are less activity. Here we are looking to the migration cell number, and we can see that the well analysis described that migration and invasion capacity in these 2 cell lines were markedly repressed by NFX silencing. And the last one, we are looking to the apoptotic rate, and the flow cytometry show us that circular RNA inactivation result in high apoptosis production. So I think this is a good example to show us that there is hope for the patient if we are looking to the circular RNA in the blood, like and tissues for biomarkers and maybe treatment. So in conclusion, this is the different function of circular RNA in the malignant phenotype of glioma. I talk you about the proliferation, invasion, metastasis, angiogenesis, metabolism, and apoptosis. The circular RNA has an impact on the promoting effects, but also in the inhibiting effect. That's why it is interesting to work to play with the level of expression on the circular RNA. So glioma is linked to the aggressive cancer of the central nervous system, but there is a difficult early diagnosis and unfortunately relapse after treatment because of the involvement of circular RNA in cancer aggressiveness. But as I said, it is interesting to use circular RNA for the early diagnosis in blood and plasma, thanks to their stable organization and for personalized treatment. So we can find these circular RNA in tissues, cerebrospinal fluid, blood, and urine in order to do diagnosis, prognosis, maybe to see if chemotherapy, radiotherapy, and immunotherapy are working for the patient. So we can take a little bit of blood. We are looking for the circular RNA, and we can see if there is a good impact in stopping the proliferation of the glioma cell. Thank you for your attention. So we want to apologize to you because we did a mistake when we took this article. It was not the good one. So this morning we tried to do the good one. So we are going to show you the other article.
So, the second article, which titled a novel integrated system using fashion with glioma cerebral organoids and developments for disease modeling and drug screening. First of all, glioblastoma multiforme is the most common and aggressive primary brain tumor. Is more 75% of primary malignant brain tumors. Despite therapeutic advances, the prognosis remains poor with a median survival amount of 15 months and an overall survival rate of 2% at 5 years.
We can propose to the patient the surgery, which is very invasive. We can also propose chemotherapy and radiotherapy. And most of the time, the doctor proposes to the patient the thesaudomide, which is a chemotherapy to the patient. But there is a lot of relapse even with those treatments.
For the problematics, the aim of authors study is to develop an integrated system combining fashion-derived brain organoids and neural xenografts in order to better understand disease progression and improve the efficacy of the therapies. Researchers have developed an integrated system combining fashion-derived brain organoid and xenografts model to model glioblastoma and test therapies. This model reproduces the brain environments and the tumor characteristics, providing an innovative platform for studying disease progression and evaluating therapy.
So for that, the researcher isolated cultured glioma patient-derived tumor cells from freshly harvested surgical tissue from glioma patients in Zianda Hospital Neurosurgery Department. So they took the cells from the tumor and they do a culture. Then they do a mechanical tissue dissociation, then trypsinization, which can release individual cells. They remove all the erythrocytes in order to do tumor cell culture. In order to obtain GPDC culture that retain the molecular and transcriptomic characteristic of the original tumors, we will see that in the results. They work on 2D culture in 3D dimension, and they do xenograft on nude mice. So for the first figure, autopic
We can't see your
models.
presentation online. Your slides are not moving.
Perhaps you have to shut off your
No,
micro. No?
it's like this one.
Ah,
You're not sharing your slides.
ah oui, pourtant il est marqué que je partageais.
Oui, pas l'écran.
Network.
Okay, it's fine now.
So the first results of this article was that autopic GCD models summarize clinical symptoms, tumor progression, and patient survival. You can see, I don't know if it works. With this MRI of intracranial xenograft, 80 days after the injection on the mice, you can see that there is the development of tumor. So here, such as ventricular compression or peritumoral edema. They also try to look at the hemiplegia in mice, and thus this mouse model showed the occurrence of a specific clinical symptoms in xenograft mice, indicate a tumor progression. And they look at the survival curve, and we know that, and we see that this orthopic model show a notable difference depending on the grade of the tumor implanted. So you can see here that they are different survival period depending on the injection of the tumor. For the second results, they show us that GPDC implantation in a brain organoid model show glioma characteristic in differential sensitivity to the tubulin. So for that, as I said, they took the GBM tissue of patients. Then they took all these cells and they put and they put these cells on the cerebral organoid they did before, and then they put a tizolomide treatment in order to look at the mutation or the resistance that can occur. Unlike 2D culture, organoids offer a context there where tumors and environmental interactions strongly influence treatment resistance, highlighting their usefulness in for more realistic testing. And that's how the results. There are variable rates of survival and apoptosis depending on their sensitivity to treatment in organisms, but some cell lines here for the 7 and 4 show resistance in organoids, because maybe due to the influence of the three-dimensional environment that protects tumor cells. That's why these results underline the importance of testing treatments in complex systems to better predict clinical responses before giving treatment to patients. And for this type of results, they look at the integrative system of patient and xenograft-derivative glioma brain organoid for disease modeling and drug screening. And they see that for the patient C4, there is a mutation here in the PTN gene that we saw in the last presentation. And PTN is a key tumor suppressor gene whose mutations are associated with aggressive tumor behavior. PTN play a crucial role in regulating growth in the immune pathway, and it does promote and control tumor cell proliferation. In another figure, we saw that KEF, which is bioinformatic analysis, revealed that GPDC exhibits marker activation of the P3K Akati and MAC kinase, also red pathway, all associated with tumor proliferation, survival, and invasion. This result confirmed that integrated model can reproduce the molecular signature of human gliomas, paving the way for the identification of new treatments. And for the last one, they look at the genomic and transcriptomic characterization of GPDC. And we can see that there are different molecular signature. For this one, here you have the glioma, and here a normal patient, and you see that there is an augmentation of the gene's expression of GBM, and this signature is strongly associated with a poor prognosis for this patient.
To conclude, there are crucial advantages like researches, developments to personalized treatments, perceiving tumor heterogeneity and the cerebral, and and a more accurate representation of tumor complexity and comprehensive analysis of therapeutic response.
And that's right.
Thank you. Any questions?
Yes. First of all, congratulations for your presentation and that you did it this morning. I think it was really good. And I just want to know how did the protein culture the organoids of the brain? Do you know that or no?
Can you repeat it, please? I didn't hear it.
How did the protein culture the brain organoids? What did they use?
Okay, so we use it was in the material and method, and I read it very fastly.
Yeah, I understand. If you don't know, it's okay.
So I think they use the project too in order to construct them. So they use, I think, the progenitors for
You have it on the slide.
yes, it was. It was the G. Yeah.
On this one.
Yes, yes. It was the G from the tumor.
Just but for the organoids. Because
Oh,
you see, we
yes,
have
for
not-
the, yes, for the,
That's your question, yeah.
it was 30% lethal, yes. But we don't know if this cells comes from which patient.
They do not come from any patient. So
Okay.
we can buy IPS. So they use a IPS cell line.
It's just a GVM tissue that is from the patient.
Yeah.
Yes.
But they yes, they produce cerebral organs. From the IPS, there are some specific organ cells in produce cerebral organs. Some time ago, we called them mini brain. Yeah. But today it's not cloud even more. So it's
Here it was just a base cerebral organoid, and then they had just the GPDs implementation.
they engraft the glioma cells.
And they I don't know, they connect properly in the environment of the cerebral organoid.
I connect. They put them really in the organ, not in the material around the organ. That's your question? Yeah, no, they engraft really in the organs. This model is introduced, but is used in Toulouse, for example, in the team of Alice Davy, where they work on cerebral organoid. They use neuroblastoma stem cells, more initiating cells, and they do something and graft the cells in mice or in organs.
I'm doing.
For
that.
example, but overlap of
Yeah,
so.
but I'm doing that in the lab, but just glioblastoma organoids.
Okay.
So I don't know if this method should be several organoids and put glioblastoma for example.
Where do you?
Yeah, that the team in the university.
Okay. Other question, yeah?
I have a more general question. Do you know if there are teams trying to modelize the brain-blood barrier in the organoids?
There are some papers on this, but I don't remember if they are really succeeded in reproducing with brain blood.
It's a very important aspect for
Yes.
after
And
reproduce clinical
yes, and for
Right.
therapy, it's also really key.
limits of these some
Yes.
organ models in this specific organ.
Other?
I have a question. So, in the first paper, I wanted to know, I think I didn't mention it, but was the
We
first
don't really, sorry, we don't really hear you. Can you ask the question on chat, please?
Yeah.
Thank you. While we waiting, do you have any other questions without?
But why not?
Can you think about the on the metathesis and on the material associated crystals? Oh, in the sorry, in the first one, circular RNA.
It was a culture not in vivo, it was the in vitro. In vitro, it was an in vitro culture with these two types of glioma. They come from two different patients, but I didn't really take my time to explain all the article because it was just for illustrate my results. It is good for you.
Things? No? Okay, so I will put my computer. Thank you very much.
Thank you.
I look at that and I next job I want to see down 1 hour. We have family.
Since we have already this. Est-ce que c'est mieux là? Pas du tout. En Zoom, est-ce que vous m'entendez?
Oui.
Ok. On va voir si ça marche comme ça. Ça va pas être possible. Et là, c'est toujours bon? Est-ce que là, c'est bon? Vous m'entendez toujours?
Oui, oui, oui.
Ok, et là, nous, on n'a plus d'écho. C'est bon? And you see the PowerPoint? It's okay? Is it okay? It should be okay for the screen share on Zoom.
Yes, we can see.
Ok, thank you. Alors, moi, j'ai le chat ici. Parfait. So, the purpose of the two papers was to introduce the use of organoids in cancer research. Of course, you already heard about organoids, I'm sure. Some of you work already on this. Many of you work on organoids during their internship. 1, 2, 3. Okay, so today it's, as I already said, it's more and more general. But I will just introduce the problematic of 3D models, so as preclinical models. Sorry, there's a mistake in the title. But because there are some specific aspects associated with this application. So the development, okay. So the starting point of the 3D models is really to have models that are more predictive of the response observed in patients. So the idea to have conditions for culture that are closer to the physiology that exists in vivo, the final point really is to be more predictive of the response. When there is a development of a therapeutic molecule, it is first tested in the tube to see the interaction with the other proteins or with the substrate, for example. Then it is tested on 2D cell cultures. And at that point, there are some results that lead to the choice to continue research on this molecule or not. And the problem is this one. Is really the time and the cost associated with the development of a drug. So here, primary screen is really the biochemical function. Secondary screen, here we are on cellular models or animal models also. And then there is here a go, no go decision to put the drug in phase 1, 2, and so on. Okay. And at the beginning, it's an idea, but at the beginning, with 10,000 compounds, it's necessary to perhaps have one FDA approved drug at the end. with quite 10 years, okay? And this process is really expensive, as you can see here, the cost, the final cost. So the problem is really this go no go decision, because at this point there are many molecules that will go to phase 1, phase 2, phase 3, but that will have no effect on patients. Okay, there can be it's possible that some toxicity problem occur. This is some reason to exclude a drug, but in many cases, the problem is that the molecule in patients has no effect, who is really, really less efficient than in preclinical tests before going to phase 1 and 2 and so on. So the objective is really to have more predictive model, to have a better selection here of molecules that will be selected to go to phase 1 and so on. Okay, so that's a very strong concern around the use of 3D models in general in cancerology, for example. So the place of the 3D models in this system, it is not really recent as a review, but it is still true. And Kunchunga, Leonie Kunchunga, was one of the first after Minabicel to try to place 3D models in this process of evaluation of molecules. And so it is still true, 20 years after. So after the discovery of a target, so there is primary screening, okay, with biochemical assay, cell-based assay, then secondary screening with animal models. And the place of the 3D models are in between cell-based and animal models today. And it was already at that time proposed that 3D models can be used in place of animal models. So this is more difficult to accept in an ethical point of view because of the problem we already discussed during the presentation of the publications. So there's only a part of the tissue of the organs which are represented, which is represented in 3D models. There is no vascularization, there is no immune system. We already mentioned all these aspects. But last year, for the first time, FDA approved a molecule that has only been tested on organoids and not in animals for a specific pathology. Okay, so that was a really strong announcement at that time. Is it a little bit special because it's a repositioning of drug? So the toxicity evaluation has already been done before. So all the toxicity aspects was evaluated before, okay? But for the pathology, only organoids have been accepted by the FDA to approve the drug for a specific treatment. So we are perhaps moving from this place here to this place really in parallel or in place of animal models. So this leads to some challenge concerning these 3D models. Because when you want to test a drug on cells, you really have to test it by multiple concentration, multiple cell lines, with high reproducibility between experiments, between cells, between models, and so on. So to do high throughput screening. And today, spheroids are spheroids can be used for high throughput screenings, and they are used. But organoids are not yet used for high throughput screening. And the main problem is the reproducibility of organoids and the cost of organoids production. So when we speak about these 3D models, so we heard about spheroids and organoids, the first presentation. They are the two main 3D models today, and but there are also 3D printing, tissue engineering, isolated organs and tissue that can be cultured ex vivo. Example in tumors, for example, there are tumor slice cultured ex vivo that are also used or also developed in a way to be able to perform screening of molecules on these models. So this is also 3D models. And so in parallel, there is animal models. About animals, you see here low productivity, high cost, social and political pressure. You heard about free air problematics, and now we hear about specific taxes on animal models. So it is not yet admitted, but it is in the corridors. And the low productivity is more surprising in this scheme. But the point is that animal models are interesting in the way they are vascularization, there is an immune system, so all the physiological systems are there. But for some tumors, for example, the architecture of the tumors, the cell-cell interaction, the density are completely different from the human. And some aspects are more are better reproduced in 3D models in vitro than in animals. So this is dependent on the type of tissue or tumors you are working on. So as I said, there are some different types of 3D models, different type of culture systems with microfluidics. We heard about this culture system in organ on chip aspects. You all heard about organ on chip already? You should. No, not everyone. So organ on chip, the idea is to have like an organ, but on a slide, on a microscope slide. So there is some like holes where you can put cells or organoids. More and more it's organoids with channels but in between that make the relationship with flows, with fluids and flow inside the different part that really exists in an organ. And on chip, because it's really to have a small sample that can be used on a microscope on whatever you want. When you see here scaffold, material is a scaffold, for example, it's matrix. And about this scaffolds, so material is most used today, but there is a development of synthetic matrices also that are also more and more used because they are less expensive and more reproducible in their composition. And after the idea to perform free mouse. Here, here, here. It's okay. So this one shouldn't work. No. So when we speak about preclinical evaluation, we need to adapt 3D models to the screening systems also. So we must be able to put 3D models in different imaging systems, if imaging is the way choose to evaluate the molecule, to perform informatics, bioinformatics analysis, perform RNA extraction, and so on. And there is also often phenotypic screening application of 3D models. I will go quickly on the next slide because we already speak about this problematic of the predictivity of the model and the way the idea that these 3D models are developed to be more predictive than 2D models, but also than animal models. So here is just an illustration to see that when we speak about tumors, for example, here, in a tumor, it's not homogeneous at all. It's really heterogeneous. And here, if we look at this part of these images here, this can be reproduced by a simple spheroid, for example, this kind of organization. But we need to have extracellular matrix. In these extracellular matrices, we will find tumor-associated fibroblasts, we'll find immune cells, and so on. Okay. So this is the same idea here, where we have here mammary tumor, breast adenocarcinoma, with the surrounding tissues. And tumors today are considered as organs and not only as proliferative cells. It's really heterogeneous. We just speak about gliomas. Gliomas are glioblastomas. There are some stem cells, there is some differentiated cells, there is some proliferating cells that are not stem anymore. So, and this heterogeneity is really characteristic and contribute to the resistance to therapy. And when we want to test new molecules, it's really interesting and important to integrate the cell heterogeneity in the models used to do the evaluation and to be, again, more predictive. So we already discussed about all these different kind of 3D models. So we speak about organoids and a little bit about spheroids. Spheroids are very simple. Spheroids are tumor cells aggregated together, but they, as it has been said in the first presentation, there is a kind of auto-organization, by the way, because it is not loose at all, the organization in a spheroid. Cells are really interacting with each other with cell-cell adhesion, and tensions also that are developed as the spheroids grow, and that participate to establishment of proliferation gradients, nutrients gradients, oxygen gradients that participate to the heterogeneity of cells inside the spheroids. Patient-derived xenograft, we heard about this during the presentations, with in some cases, it's directly the cells that are engrafted in the mice, and more there are cells from the patients are used to do 3D models like organoids that then are engrafted in mice. So different type, different technology. So this is a model we heard during the presentation with the engraftment of glioma cells in a printed. In this example, it's a printed brain organoid. So bioimpression of cells. You know what I'm speaking about? Yes. Okay. And so there are many publications about development of technologies more and more fancy to produce 3D model more and more complex. So at that time, from my point of view, we are still in a strong development period about these 3D models, and not completely in the application of all these kind of 3D models in evaluation. It will be in some years, but it will be very efficient. Sure. Okay, this is not, so just some word about the spheroids, because this one, you do not hear about it. No. So this is an image of a frozen section through 400 microns in diameter of spheroids. So we can produce spheroids of half a millimeter quite very easily. So this is, in this spheroids, there is cell heterogeneity. We reproduce a kind of 3D architecture, cell-cell communication, and it is possible to put spheroids in matrix, and so to have cell-matrix interactions. So this spheroids models are, it is possible to produce spheroids made of different type of cells. So we can introduce in these spheroids cancer cells, of course, but it's possible to mix tumor-associated fibroblasts, for example, to take into account the impact of this tumor-associated fibroblast on cancer cells in the evaluation of molecules. There are some models where there is also some immune cells that are being mixed with the cancer cells to produce the spheroids. So there are some different models with different kind of complexity. As I said, there is some gradient here of proliferation of oxygen and nutrients, leading to the presence here, for example, of a core of quiescent cells. And in some models, we can also have a core of dead cells, as it exists in some tumor. For example, in mammary adenocarcinoma, it exists sometimes to have a specific, in the center, some dead cells. So this is possible to reproduce this in large spheroids. In this model, there is also the presence of physical barriers. This is important for the diffusion of molecules. So these physical barriers are represented by the extracellular matrix that can be deposited around cells. The cell-cell interaction also. When we treat spheroids with a molecule, very often we really see a strong effect on the most peripheral cells, and very few effects in the center. And gene expression level, there are some studies that have compared gene expression by transcriptomic approaches between monolayers and mice for different models. And most often, the expression, gene expression in spheroids is representative of what is observed in vivo, and really different from what is observed in monolayer cells. So it's already, this simple model is already a gap between the 2D cells and more complex physiological conditions. And the growth of spheroid, because during the culture, this structure will grow. And in the first phase, there is a rapid increase in the volume. A quick increase is here in the volume that can be considered as an avascular growth of tumors. And then there is like a plateau. And this plateau could correspond to the time where in tumor angiogenesis should develop, but that does not exist in this model. And this simple model of spheroids can also be used to test molecules on invasion. To when people or lab try to study the metastatic pathways, we can test very simple and quantitative assay using spheroids to evaluate impact of molecules on invasion. I just mentioned the aspect of quantification. This is also an important aspect when we want to evaluate molecules, because it's we cannot just say, oh, it's less big or perhaps more big. It's not enough, of course. So it is really important when we want to do preclinical evaluation on 2D, but it's also important in 3D models to have quantitative assay to be able to quantify the effect and then to compare the effect depending on concentration on different experiments, and you know all the pipeline. So okay. To make spheroids, it's really easy. So that's why this model today is used in the industry to test molecules. And that's why in research, this model is completely has-been. Because it's, and it's absolutely normal, because it can be used to evaluate, but it's really important to have more complex system, of course. So the first method here, the Angie drop method, is not used anymore. It was the first method. It was not so easy, and forget it. Liquid overlay is used with some system, depending on the cells. It is necessary to have some also agitation, like rotation and this kind of things. And the more used method today is the use here of multi-well plates with round bottom wells, and with the wells that are treated for cells not to adhere. The way to produce spheroids is really to avoid cells adhesion on the substrate, on the culture plate. And then cells will adhere with each other. So this is the mostly used today. Then we can put cells in the well, use only a simple centrifugation on these low attachment plates. And then if we follow here the progressive aggregation of the tumor cells, we can see that at the beginning, in few hours, the structure is quite loose. And if we pipe and this, cells will disperse. And in one day, we have really well-organized, auto-organized spheroids. And this is clearly identified by the regular border here, that represents the tension between cells due to cell-cell interaction and cell-cell adhesion. So just an illustration here. where we can see the cells aggregate to each other, and then progressively they compact to form a dense and organized structure. I show it there because, as you see, it's a simple video microscopy in transmitted light movie. So we put the plate on the microscope, inverted microscope for live imaging. We did an image every 5 minutes, something like this. And then we analyze the image. And on this kind of thing, we can directly do automatic image analysis. That is represented by the red line around the spheroid. So this is the result of automatic image analysis. And as soon as we have this automatic image analysis, we can perform quantification. For example, the evolution of the area. And this kind of, then from this quantification, for aggregation, for example, we can quantify the decrease of the area. And we have this quantification for 96 spheroids at once. And then we can test, for example, the addition of molecules on this. And for example, in this work, we used this first step of spheroids formation as a model to reproduce cluster of formation of cancer cells that are found in circulation of patients. And then as these clusters have more metastatic potential that is higher than individual circulating cells. And then from this kind of assay, we can try to find molecules that inhibit the formation of cluster, and then that can be used in therapy. Okay. This is not really interesting, and it is not used. I haven't put it. Yes. I find my mice. Okay. Just to illustrate the problem of high throughput screening using these models. In the kind of development that have been performed, and that are this kind of development are now used for organoids. Here it's so in a classical way, we can obtain one spheroid per well with on one plate an homogeneous size of the spheroids. It's less than 5% of variation in size in one multi-well plate. So this is important in terms of reproducibility of the evaluation. But if you use spheroids in extracellular matrix, for example, spheroids will be at different level in the well. So to use imaging, you have to perform 3D imaging, so it's more complicated. And they will have some variation in size. So to go in this, improve this aspect, there are some development that have been based, on the development of microwells in PDMS that are put in wells of plates. So to show you here an example, so in this example, they were able to have 700 microwells in a one well of 96 well plate. So 700 spheroids in one well. So to test one condition. This the increase, of course, the efficacy in high throughput screening. And using this kind of development, they tested doxorubicin, for example, another drug here And then they can, using live dead staining, evaluate the response to therapy directly in the plate, in the well, on a high number of samples per condition. And here, again, they find a way to quantify the response. So in this example, they use the percentage of spheroids. They could have been used the intensity of annexin 5, for example, of the intensity of propionate iodide to detect dead cells. But it is important to have a quantification to perform evaluation. And today in organoids, this is also a limiting point to have them integrated in high throughput screening for preclinical evaluation is really to have this quantification. So really assay based on quantification of the response. It is not yet in place. So I really have to stop. So this is a product that is commercialized, for example, and you see it's really clear multi-well plate, and in the plate there is nano-pattern surface, and when the cell there are some spheroids. So these things will be used with organoids, it's sure, but the culture organoids needs more and more development. It is really important. So organoids concerning the 3D models today for preclinical evaluation, with the idea of screening of molecules, it's more for 3D models, spheroids, and organoids are hopefully used in research, of course, and there are many works to apply them in high throughput screening, but it is not yet completely possible because of the reproducibility and the problem of quantification of the response to the treatment. Okay, that's the main messages I wanted to give you. Do you have any questions? What's clear? Yes, I will put the slide on Moodle. Okay. Thank you.
Change something. I think my name is not on the attendance list.
Okay.
I don't know why, but we didn't say my name.
Do you have any Zoom? Sorry, do you have any questions? I will show you by the rules. Okay. So you want to check? You are in the care?
Yes. Yes, here.
Has it?
Yes.
Okay, so you're there, and it's okay. I put your in. Isabella, Yamina est là. Marianne est là. Là, quand je n'ai pas là, Myriam, c'est bon. Diazana, elle était là, elle était là. Robert Sophie, Chaudet, Hélène, Malma, c'est bon. Yamina, c'est bon. Basile, c'est bon. Ok. Okay. Thank you. I will quit the Zoom. Okay. If you have any question, do not hesitate to ask by email. Okay.
There is another course, but I can take it.
Thanks. Au revoir. Merci.
Something like that. First, genes. I don't remember the